quorum sensing, let me just make this disappear for a second. Quorum sensing is a major behavior that bacteria need to navigate their world, because they often need to figure out whether they're alone or in a group. And again, if we think about what senses do they have, they don't have sensory organs, and they don't have neurons, and so they have to figure everything out using simple chemistry with some enzymes, some receptors. And so this is an elegant set of systems that they can use for that purpose. So the name comes from quorum, which is a word you see in committees. When a committee meets to make a decision, usually they're only allowed to make a decision if enough committee members have showed up to the meeting. And it just makes sure that two committee members can't secretly meet somewhere and make a decision on behalf of everybody. Instead, you have to have enough people there to make a formal decision. And so what they say is, do we have quorum? And that is, do we have enough people to make the decision? And so quorum sensing is when bacteria are asking, do we have enough of us here to do a group behavior instead of the individual behavior? And I typically want you to think about, why would there be group behaviors versus individual behaviors? Well, we see this in humans, where we have this silly example of Homer Simpson looking like a maniac, trying to tell everyone something. And people would be looking at him thinking, okay, well, there's the maniac with the sign. No one pays attention to him. But if you see people at a protest, they all feel powerful and they can't really be ignored because they're in a large group. It's a basic thing about our psychology. Okay, so the first example of quorum sensing I'm going to give you two, and I'm going to walk you through this one in the most detail, is the example with Vibrio fischeri in Eupremnus galopes, the Hawaiian bobtail squid. This is an amazing example of a mutualistic symbiosis where this squid is a nocturnal squid, and it's going to hunt at night pretty close to the surface of the water. And it needs the bacteria because when the moon is up, anything below this squid looking up at the surface of the water is going to see this silhouette. And so a predator is going to see a silhouette and eat the squid. So the squid needs to somehow hide itself and get rid of its dark shadow. And so what they do is this squid has a special organ that feeds nutrients to these bacteria to get them to glow. And that kind of makes this less of a shadow and camouflages the squid. So what you're seeing in these pictures is this same petri dish in normal lighting, where you can see what color the cells are. They're yellow. The colonies are yellow. And then if you turn the lights off, the light they make is blue. So it could kind of look like moonlight when viewed from underwater in the ocean. So that's what the bacteria are doing. That's the kind of behavior they're trying to control. And the idea is that making light, as you will see, it is an energy-intensive process. And if there's only one cell present, the amount of light it makes will be undetectable. And so it's going to be using all this energy to make a pointlessly expensive, invisible amount of light. But if it's with 1 million of its friends, if you have 1 million cells together, the light they make will be visible and the squid will be able to use it. So the squid is paying attention to how much light it's getting from the bacteria, and if it's not getting enough, it'll flush them out. So that's a complex squid behavior. But the bacteria have to figure out if there are enough of them to try to make the light. And so that's what they're going to use the quorum sensing for. So one thing we have to understand before we get into the quorum sensing is operons. So this is a thing from bacterial genetics. So what is an operon? I don't know if you covered this or not, but in your biology class, you might have covered the lac operon, which is an example of genetic regulation in E. coli bacteria. And what any operon has is a set of genes in bacteria. This is a bacterial thing. A set of genes that share a single promoter, and they are transcribed onto a single messenger RNA. So they are really right next to each other in the genome, and if one of them is transcribed, they all are on the same mRNA molecule. So the idea typically is that the bacteria want to have several related enzymes made at the same time. They say, oh, my environment has changed. I want to turn on a new bacterial metabolic pathway. All those genes are on an operon, turns that on, makes all those enzymes for that new pathway. And this quorum sensing thing is another example of an operon where the light-making apparatus is several different genes that are part of an operon. So an operon is a group of genes that share a single promoter and are transcribed as part of a single messenger RNA. So then what's the next step after you have a messenger RNA? Well, then the cell can translate the genes and synthesize the proteins. And this is where, even though the genes are all in the same RNA, they can have sort of different translational regulation. So some of the genes, you'll get lots of copies of a protein, and others you'll get a few copies of a protein, even though they're all on the same messenger RNA molecule. So you don't get the same numbers of every protein, but they all get made at the same time. That's the idea. Okay, so let's look at how this system works. This is the way we draw, or we look at genes in bacteria. We'll typically have an arrow. So this line here is meant to be part of the bacterial genome, a very small part of the bacterial genome, the part we care about. And this arrow represents transcription of this operon, which is going this direction. So that's on whichever strand of DNA would be transcribed in this direction. And this arrow going the other way is a different gene that's on the opposite strand of DNA, so it's transcribed in the other direction. And so the way we make, the way we name genes in bacteria is with lowercase, usually a 3-letter designation that tells sort of what it does, and then we give it a letter. So how to read this is LuxI. Lux are the luminescence genes. So LuxI, LuxC, LuxD, LuxA, LuxB, and LuxE. They're in no real order. The names, at least, don't tell us much about the order. And that's because we discovered the genes before we discovered how they're organized. So if they had been discovered through gene sequencing, they might have been named A, B, C, D, E, but they're not. Okay, so in this system, we have an operon with all these genes that are going to be transcribed at the same time. And then we have a separate gene that's going to be transcribed separately. So they're going to be regulated independently. This is not going to be transcribed at the same time this is. So what do these all do? Well, Lux C, D, A, B, and E are all involved in making the proteins and precursors that are ultimately going to make light. And this is an energy-intensive process. It takes a lot of proteins and it takes a lot of metabolic activity. And so the cell never wants to transcribe these genes at the wrong time. It never wants to make light for no reason. So that's why this whole system exists. So what else do we have? So Lux, C, D, A, B, and E are the machinery for making light. We also have LuxR. This is the receptor, so that R does mean something. LuxR is the receptor protein. So what is it going to detect? It's going to detect the autoinducer. So LuxI is the protein that's going to make the autoinducer molecule. And, excuse me, notice this is capitalized where this is not. And that's how we designate this is the name of a protein where this is the name of a gene. So LuxR, lowercase lux, that's a gene. LuxR, capital lux, that's a protein. So this is what we call the autoinducer gene. It's going to make the autoinducer molecule. So also, when are these going to be transcribed? What has to happen to make them transcribed? Well, what has to happen is this LuxR has to bind here in a certain condition. It's going to bind here and initiate transcription. It's going to recruit RNA polymerase to initiate transcription. So this molecule, once it has detected the autoinducer, it will bind here and make transcription happen. Also, there's always going to be a very low level of transcription of these genes with translation of LuxI and not much translation of the rest of them. The cell is always going to make a little bit of the LuxI protein. So the LuxI protein is going to make the autoinducer molecule that I'm going to draw as a square. So that is called an acyl or acylated homoserine lactone. So this is a lactone, and somewhere in here is the amino acid serine as part of this structure. And this structure doesn't matter. I'm not going to ask you to draw it, though I am going to ask you to think about its chemical properties later. So the cell is always making some small amount of this protein, and it is always making some amount of this molecule. And that molecule can diffuse across the membrane, because if we look at it again, it's moderately hydrophobic. It's not charged in any way, and it has a lot of hydrocarbon stuff here, so it has lipid-like properties, so it can diffuse across the cell membrane. And so the cell is always going to be making some, and it will always diffuse from higher concentration inside the cell to lower concentration outside the cell. That's what it will do. If the concentration inside the cell gets high, if there's a lot of autoinducer inside the cell, it's going to bind to the LuxR, to the receptor, and that's going to activate much higher levels of transcription of this operon. So it's kind of this loop where this protein is going to make the autoinducer, and if enough of the autoinducer is present, that's going to cause more production of the autoinducer. So this is it's a possible feedback loop. So let's think about what can happen. So if this is a cell alone by itself, it's going to make the autoinducer at a low level, and it's just going to diffuse away from it. So it's never going to achieve a high concentration inside the cell, so it's never going to activate, it's never going to ramp up transcription, it's never going to bind to the receptor. And that's good for the cell because making light would be pointless and energy intensive. Contrast that with if you had a bunch of cells together, well, they're all making the autoinducer. So for a lot of these cells, the external concentration of the autoinducer is higher than the internal concentration. So it's going to diffuse in, and it's going to make a higher concentration that's going to bind to the receptor and ultimately make more transcription happen. It's going to make more autoinducer, all these cells are going to get activated. They're all going to start transcription of the Lux genes, the light-making genes. And that's what this is showing. And that does become a feedback where once some of these cells are activated, they ramp up production of everything here, including the autoinducer. And so that just vastly increases the amount of autoinducer present. So all the cells end up getting activated. And so if in a large enough group, quorum will be achieved. And what that leads to is they will all start producing light. Okay? So that is the Lux system of quorum sensing. And this is a slide where I write that all out for people who didn't watch the videos or people who watch the videos and want to go through this later. You do have to spend some time on it to understand it. But again, this is a good example of the way a bacterium solves a problem, the way it sees its world using a small number of receptors, a small number of proteins and genes, allow it to do something fairly complicated, which is figure out if it's in a big group or not. And one of the goals of a microbiology class is to get you to think like a cell, and this is a great example of that. So that's the Lux quorum sensing system. And there are other systems out there, and I will talk about them in the next video. So I'll see you there.